Okay, thank you. I wanted to talk about our use of using custom arrays in the lab to complement our NGS testing and how we've used the medical research exome array. There. So we've had arrays in the lab for quite a few years, and our process initially had always been design the array for the content we want for the specific gene panels, then evaluate that design, and then validate it. So our design could come from a commercially available design. It could come from a design from a list that we handed to a company of the genes we were interested in, and they designed it for us. Or we could use the tools that were available to design the content ourselves. But either way, that design had to be evaluated and validated before it could be put to use in the clinical lab. And that would be for a couple of panels, or arrays or tests that we were interested in the lab. And each time we wanted to introduce new tests, we would have to go through the process again of design, evaluate, validate. So this was a very reiterative process to get new tests into the lab. It was time consuming. So when OGT introduced the mRNA in October of 2014, we thought of this as a way that could address our problem, not by introducing the 1 by 1 million mRNA chip into the lab, but by taking that content, evaluating it, validating it, and then having that resource of 4,600 genes validated internally in our lab to pull from to quickly introduce new arrays into the lab, new array designs using sub-content from the 4,600. So as was mentioned, this contains medically relevant genes, and very importantly for us, it's able to detect single exonic level variations. So about 4,600 genes, the goal was to have a minimum of 4 probes per exon in the design. And that's what we wanted to base our calls on. And we wanted to be able to feel comfortable that we could detect a single exonic level change. But not all genes are equal. Here's an example of how the vast majority of the genes look on the array. They have nice spacing across the intronic regions, targeted in-depth coverage of the exons, and then a small flanking area. There's also the backbone region of the chromosomes targeted. But not all genes are perfect genes. Not all genes cooperate biologically. As you see here, there's the various transcripts shown, but the row above that where it says user-supplied probes shows the probes that were actually able to be placed in the design. And these genes, because of those segmental duplications, their similarity elsewhere in the genome, they were not able to place unique probes into those genes. So there were a total of 36 genes out of that 4,600 genes that when we examined the list, we said there's just no coverage there. An attempt was made, but no coverage. And then, like I said, we wanted to make sure that we were covering, that we had a set of validated probes that were covering the exon at the exonic level. So for us, it was important that 70% of the exons in any given gene be covered with 4 or more probes. And not all genes were. Again, you can see segmental duplications cause a problem with placing unique probes. And in this example, with the GSTA2 gene, 6 of the 7 exons only have 3 probes. So they were able to get some unique coverage, but not enough for our calls of 4 probes to detect a single exonic level change. Here's an example of another gene, the PRB1. So the reference transcript is the one in blue there at the top with 4 exons. And again, only one of the 4 exons were they able to place 4 probes in. But this was only 96 genes. So a total of the 36 and then 94 genes that we didn't have coverage at 70% or greater level of the exons. So really just 130 genes out of that 4,600 targeted genes didn't meet our criteria. So 97% of the genes had that excellent depth of coverage that was shown at the beginning for the ACTA2 gene. So when we subtracted those 130 genes that didn't meet our internal criteria, we were left with approximately 6,300 exons that had been targeted for coverage with 4 or more probes. And of those exons, almost 95% had at least 4 probes in them. There was a small percentage of genes where they were covered at 70% of the exons did have 4 or more probes in them, but there may be a sporadic exon here or there that did not have 4 probes, and we felt those were important to know about and to be able to list as limitations. So when I say they don't have 4 or more probes, that doesn't mean they're always significant. For example, you could have a UTR exon that doesn't have 4 probes. Here it only has 3, but because we use our arrays in combination with our NGS, and we want to target primarily the coding regions on both of those assays, the lack of 4 probes in the UTR is not significant for us at ARUP. So this is an example here. Another example is an alternative transcript. Our NGS test targets specific transcripts. So the arrays, while targeting all transcripts, if that uncovered exon, low coverage exon, is an alternative transcript, then again, it's not significant for us in the design. Sometimes the same problems that led to a gene not being covered leads to a few exons not being covered, such as in the CHEK2 gene. So again, we have segmental duplications indicating similarity elsewhere in the genome, here on the 3' end of the CHEK2 gene. And probe placement was not good in 3 of the 15 exons. So coverage was attained for 80% of the exons, but we have 3 that had almost, but not quite. We couldn't make a call for a single exon deletion for 3 of the exons of CHEK2. So we just feel like this is a limitation that we need to know and be aware of. Here. So of those 63,000 exons, with the 5% that had low coverage, 1.6% of them were in UTR regions, so insignificant to us internally. Most of them had 3 probes at least, 3 probes, so very close, and perhaps if you expand into the intron, you might be able to get your coverage, or you could go back, if that gene's very important to you, you could ask for a review of the design to see if additional probes could be placed in there. And less than 1% had 2 or fewer probes in them. And again, those were usually for very good reasons that you could not get unique probe placement there. So now that we had done the evaluation of the design and felt comfortable with it and knew the limitations and knew the strong points of it, we wanted to go ahead with the validation. So for our validation, we used 27 medical exome arrays. The data was run and collected at ARUP, Greenwood Genetics Center, and Mount Sinai Genetic Testing Center. Most of them had very small copy number previously known. 5 had no known abnormalities. For our ones that were known, we wanted to use the smallest possible because we wanted to see, could we detect down at the resolution of a single exon reliably? So those were positive cases identified by MLPA in the clinical lab. So it's shown here by multiplex ligation-dependent assay. You see a deletion of a single exon, exon 8, in the ALK gene. And then if you look at the probe coverage, I've got exon 8 circled there, very small. There are 7 probes in the array for that single exon there. So beautiful coverage. But as you get to know any array, I'm not going to show you the sensitivity or the specificity or the reliability. I'm going to show you that as you get to know any array design, unique calls that you hadn't expected start to appear. I've had this experience with every design that I've utilized. And in this case, out of 4,600 genes, or about 4,500 genes by the time we eliminated, we had 4 regions of unexpected variation that appeared commonly. And these are, you remember, these genes are medically relevant, and we're looking for variations in coding regions. So when we see these variations, it is significant that we know how common they are. So one was in the growth hormone receptor gene. 7 of our 27 cases had a copy number variation in exon 3, and that has been reported in the literature before multiple times. That exon is a disease-associated polymorphism, so you'll just want to mark it and be aware of it as you go to interpret data about this gene. MAG12, exon 22, was seen 4 times, a deletion of the coding region of the exon extending into the UTR region. Again, while deletion of just exon 22 has not been reported, the bottom 2 there on the graph showed that deletions involving exon 22 have been reported as pathogenic. So you want to just know before you do your interpretation that this is a common finding in this array. KNSL1, exons 1 and 2, variation always including exon 1 extending into a variable amount of intron 2 was seen for both deletion and duplication at a very high rate. 17 of the 27 samples had this variant. And then the last one, PCDH-A10, exon 1, which is a coding exon again. So you just need, it's only by the validation, by the wet bench work, that you get to know your array design and the things you can expect from it. So evaluating the design of the mRNA and then validating it, we now have this set of probes that we feel very comfortable taking directly from the design, placing onto smaller array designs for panels that meet our clinical needs, and putting directly into the clinical lab. So that process has been streamlined to one time of evaluate and validate. So now I want to show you some of the results we've had using targeted exonic arrays in the clinical laboratory and how useful they can be. The first case is an HHT case. It was a 59-year-old male that presented with nosebleeds, telangiectasias, a liver AVM or arterial venous malformation that required transplant, and a positive family history. He came in not through the genomics lab, but through the genetics lab, first for sequencing of the ENG gene, and del-dup analysis by MLPA was negative. Then the doctor added on aCVRL1, again negative by both sequencing and MLPA. And then it went to SMAD4. The SMAD4 sequencing was negative, but the MLPA was positive, showing a duplication of the gene, but not an entire duplication. And I'll tie this into the array on the next slide. But this duplication did not include exon 1, did include exons 2 and 3, did not include exon 4, but did include the rest of the gene. So a very unusual pattern of duplication. And we knew from correspondence that Ambry had seen the exact same pattern in their patients. So we were really curious what was going on here. So we wanted to take a two-prong approach to investigation, real-time qPCR and array. So first I'll show you the real-time qPCR done by Allison Milson in our lab. So primers were designed to amplify exon 3, which is part of the duplicated region in this patient, and for exon 4, which is not duplicated, and then 2 housekeeping genes, and using crossing thresholds, the relative copy number of the genes were examined. And you see here that it doesn't appear that exon 3 is duplicated, that they're a 1-to-1 ratio. Well, that doesn't agree with the MLPA findings, so when we looked at the array that we ran on the patient, we see just the exons are duplicated, not the introns. So this is data you couldn't get from the MLPA analysis. We're seeing exonic level duplication, not introns. Exon 1 is not duplicated. It's an untranslated region, nor is the UTR of exon 12. Neither are duplicated. And in this example, neither is exon 4. The exon 4 is only a 30 base pair exon. Very small. You can't get a 60 base pair oligo inside a 30 base pair exon. So this led to our hypothesis that we're looking at a processed pseudogene, a novel processed pseudogene in these patients. And using that information, we went back to reinterpret the MLPA and the qPCR. And the MLPA, exon 4, is so small that the probe was actually placed in the intron nearby. So again, that probe's only going to interrogate the real gene, not the processed pseudogene. For the qPCR, the primer sets were designed with one probe in the exon and one probe in the intron. Only going to interrogate the real gene, not the processed pseudogene. So the only way we could have come to this conclusion was by using an array which showed us and gave us information on the intronic level to compare with the exonic variation that we're seeing. So then we proved this with a PCR assay that interrogated both the real gene and the proposed processed pseudogene, and you can see both size products in the patient but not in the control, and when those were sequenced, the small product was sequenced, it did go from exon 3 to exon 4 directly, confirming that we were looking at a novel processed pseudogene. Now this sort of information is important to use the array and the NGS data together, because we had another patient come in, this time for the panel that included SMAD4, and the NGS data was available first. And when analyzing the NGS data, it looks like there is a very nice splice site variant, 3 bases into the intron, a G to a C. But knowing that there was a processed pseudogene now, when you look here at exon 6, you can see the first 3 bases are a GTC. The first 3 bases after exon 5 are a GT and then a G transformed to a C. So it matches exon 6. When we align the reads, you can see that it doesn't read exon 5 into intron 5, but we're reading exon 5 into exon 6. And array data confirmed this with this patient. So it's very helpful to have both and to analyze in conjunction. Next case I wanted to look at was a cerebral cavernous malformation case where 4 patients, a pedigree with 4 individuals, was submitted to the lab, all 4 of them for testing. There were 3 affected individuals and the asymptomatic mother. And our genetic counselors worked with the physician to identify a proband who was the most severely affected and to arrange for testing of this patient first, then do follow-up with the others. So this was a 30-year-old male with multiple cavernous angiomas and seizures, the most severely affected. And when he was run on our CCM panel, where there was a deletion in the CCM2 gene, which our genetic counselors could then go back and point the physician to running just MLPA analysis of that one gene on the remaining family members and show. This case was a hearing loss case, and the reason I wanted to present it wasn't because of the interesting symptoms, but because if you look at our NGS analysis and our CNV analysis, 56 versus 53 genes there. And that is because, as I showed at the beginning, not all genes are amenable to coverage on an array. You can do some interesting workarounds with sequencing. You can take it into the Sanger lab. You can do long-range PCR. There are some techniques you can use to get around the regions of sequence similarity. With arrays, it's a little harder. So that's why this particular assay has 56 genes on the NGS and 53 on the array. And this is just an example of one of the genes that's omitted, the STRC gene, where probe placement was good only in the green exon shown here, but we were not able to get unique probes for the rest of the regions. Luckily for this patient, it didn't make a difference. There was a 5.8 kb deletion, the EYA4 gene of exons 19 and 20 that was pathogenic for this patient, and then a couple of VUSs seen by sequencing. Last case is this 1-month-old female presented with very classical aortopathy. Was run our 21 gene panel for NGS and array. And the reason I point this out is because this is a little 608 base pair deletion of exon 30, 9 probes in that region, very small, very specific. It's not the smallest deletion we've seen. That would be 124 base pair deletion, APC exon 1, but that was a validation sample with a known positive. So this was is a very small, clinically unknown. And this has been reported in the literature as well. So then last, I wanted to look at all of our arrays, the last 1,000 arrays to come into the lab. So like I said, on our array designs, we put multiple assays, and in the clinical lab, they're analyzed just for that gene set, just for the HHT genes, just for the hearing loss genes, and the rest of the data is masked and not seen in the clinical lab. So I pulled the last 1,000 cases into R&D where they were de-identified and just wanted to examine how our arrays were performing overall. And as you can see, overall, we have about a 10% positivity rate, equally divided between deletions and duplications. But the interesting part's on this next graph. 21% of those findings are in single exons. You really need an array that's targeted at the single exon level. There. And then you can see there was quite a few with multiple exons and with whole genes. But 21% single exon variations. So that's why we feel, so this has been our old way of going into the lab, a very time-consuming way. We now have the medical research exome array where we evaluate at one time. We validate it at one time. We can quickly choose genes, knowing they're validated internally at ARUP, to put as new panels. It results in a time saving, a cost saving, and a very quick introduction of new panels into the clinical lab. And so with that, I want to thank all the various groups here that have contributed.